Hello dear students, welcome to our biology program. Today we are going to complete immunity in men and especially the third line of immunity. Let's begin. When we studied the immune system mechanism in men, we studied the natural, non-specific or innate immunity, in which we knew that there is three lines of defense. The first line, it was about the outer part of the body which faced the pathogen directly, and the second part, the inflammations taking place once we are injured or the pathogen infect a certain part of the skin or of the body. And the last point, it will be the acquired immunity, which is the defense mechanism that resists the pathogen. So the acquired immunity, it is a defense mechanism that resists the pathogen, and this acquired immunity is activated when the second line of defense failed in getting rid of the pathogen. The body will use third line of defense that responds to this by a series of specific defense mechanisms that resist the pathogen. These defense mechanisms are collectively called the immune response. Then, what is an immune response? An immune response, it is a series of specific defense mechanisms that the lymphocyte perform to resist the pathogen. Mechanism of acquired immunity, it takes place as the following. The acquired immunity or the specific immunity is done through two separate mechanisms that are actually interconnected with each other. These two mechanisms are the humoral or antibody mediated immunity, and the second one, cellular or cell mediated immunity. So what is the humoral or antibody mediated immunity? It is immune response of the B lymphocyte to defend the body against antigen pathogens such as bacteria, virus, and toxins present in the body fluid, which is the blood plasma and the lymph, by producing antibodies. So this is the humoral one. It is the immunity response of the B lymphocyte to defend the body against antigen, and this is antigen and pathogen, as we knew, and they are present in the body fluid, blood and lymph, and they produce this humoral one. The blood will produce the antibodies. The stages of humoral immunity, it takes place on 3 or 4 points. These points, they will be the first, the attachments of the B lymphocyte with antigen. Let's look to the film and then speak about it again. It begins by the attachment of B lymphocyte with antigen. When the pathogen enters the body carrying on its surface a specific antigen, the B lymphocyte recognizes this pathogen and enters the body carrying on it a specific antigen and this one, the B lymphocyte recognizes this pathogen, then attach to this antigen by using the immune receptors, which are called cluster of differentiation. This cluster of differentiation are symbolized by CD19, CD20, CD21. There is another one, we are going to study it in while which is CD8 and 4. They are present on the surface of the B lymphocyte. The second, what is the role of macrophage in this humoral or antibody? At the same time, the macrophage engulfs the antigen and digests it by its lysosomal enzymes into fragments. And they divide them into fragments. These fragments bend inside the macrophage to a protein forming major histocompatibility complex, which is symbolized by MHC2. MHC2, M for major, H for histocompatibility, and C for complex. Then the complex resulting from the bending between the antigen and the MHC2 transfer to the plasma membrane of the macrophage to be presented on its outer surface to the T helper lymphocyte. The helper lymphocyte TH are symbolized by the symbol TH. Activation, the helper lymphocyte activation or TH activation. This TH will recognize the antigen by the MHC protein present on the surface of the macrophage and bend to the antigen MCH2 complex. The TH bend by CD4 present on its surface to the antigen and MHC2 to be converted into active TH. The active TH or the helper lymphocyte, T helper lymphocyte, release interleukins that will activate the B cells. So the interleukins here, this type of protein, will activate the B cells. Will activate the B cells carrying on its surface the antigen bound to the MHC2. The TH or the T helper lymphocyte can recognize antigen only after being treated by macrophage and presenting it on its outer surface bound to the MHC2. So this is a very important one. We will repeat it. The TH can recognize antigen only after being treated by macrophage and presenting it to its outer surface bound to the MHC2. Then, how the antibodies are produced here. The active B cells will divide, multiply, and differentiate into memory cells and plasma cells. Plasma cells, which produce large amount of antibodies, which pass through lymph vessels and blood circulation to fight the infection. The helper cell binds with the interleukins. Look here, the interleukins is released that activate the B cells. So the B cells here will be activated, and the receptors here recept this activation, and look here for activated B cells carrying the on the surface the antigen bound to MHC2. This is final. The second phase, response effector phase. So this is the B cells here, will divide, multiply, and differentiate into memory cells and plasma cells. The plasma cells which produce the large amount of antibodies which pass through these lymph vessels and the blood circulation and fight the infection. Memory cells will remain in the blood for long period of time, about from 20 to 30 years, to recognize the same antigen if it re-enter the body, where they divide and differentiate into plasma cells and that secrete antibodies specific for the same antigen, so a rapid response will be done. Destroying the pathogen. This is the last point. The antibodies produced by plasma cells are not effective enough to destroy foreign cells, such as the cells infected by virus, because these antibodies are relatively large-sized molecules that cannot reach the virus inside the cells. In this case, the foreign bodies will be combated by T lymphocyte. So the T lymphocyte enter here to enter inside the cell because of its smaller size. The second type of acquired immunity, it will be the cellular or cell-mediated immunity. Cellular or cell-mediated immunity. It is the immune response done by T lymphocyte, which is done by the receptors found on its membranes, that gives it a specific response to antigen. The mechanism of the cell-mediated immunity. First, the role of macrophage. When the pathogen enters the body, the macrophage will engulf it and decompose it into small fragments. These small fragments bend inside the macrophage to MHC2. The antigen MHC2 complex will move to the plasma membrane of the cell to be presented on its outer surface. The helper lymphocyte Th activation, bend by the help of its receptor CD4 present on its membrane to the antigen MHC2 complex present on the surface of the macrophage. This bending will activate the Th cells. Each T cell can produce during its maturation a specific type of receptors specific to its membrane. Each of these receptors can bind to a single type of antigen. The function of the activated helper cells Th release the interleukins to activate the Th bound to the antigen to divide to form a strand of activated Th cell, memory Th cells, that last in the blood for long time to recognize the previous antigen if it enters the body another time. Also, it secretes different types of proteins, cytokines, that do the following: attracts the macrophage to the infected site in large amount, stimulate the macrophage and other types of T lymphocyte, B lymphocyte, therefore activation both cellular and humoral immunity. Activating the natural killer cells to attack the abnormal body cells like cancer cells or cells infected by pathogen. Recognize the antigen bound to the MHC2. Activate the cytotoxic T cells or TC. Then the role of the cytotoxic TC, T cells can recognize foreign bodies by the help of the receptor CD8. found on its surface, whether these foreign bodies are transplanted tissue or the antigens of the microbes that enter the body, or cancer cells, and destroys them by the following: secreting a specific protein called perforin protein, and secreting lymphatic toxins that activate certain genes in the nucleus of the infected cells, leading to the destruction of the nucleus of its death, inhibiting the immune response. After destroying the antigen, the T suppressor cells, or TS, bind with the help of the receptor CD8 found in its surface to plasma cells, T helper cells, and T cytotoxic cells. This binding will help it to secrete protein called lymphokines, which suppress or inhibit immune response or stop it. Therefore, plasma cell will stop producing antibodies. Many of the T helper cells and activated T cytotoxic cells will die. Some of them will be stored in the lymphatic organs, where they stay ready to combat any similar infection when needed. Then what are the stages of acquired immunity? There is 2 stages for the acquired immunity. Then what is meant by this acquired immunity another time? It is the resistance of the body to pathogens that infected the body before. So we added here a certain word which is infecting the body or infected the body before. So the acquired immunity resists the pathogen which entered the body another time. This acquired immunity state passes through 2 stages: the primary and secondary immune response. Then what is the difference between the primary and secondary immune response? And what is meant by primary and what is meant by secondary? First of all, the primary immune response. The response of the immune system of a new pathogen. This pathogen entered for the first time. It is a new one. Then the B and the T cells are responsible for the immune response to the pathogen and attack it until it is destroyed. What is a secondary response, immune response? It is a response of immune system to the infection of the same disease again, another time. So the primary, it enters for the first time. The secondary, it resists this disease when it comes to the body another time. The secondary immune response, the memory cells are responsible here for this secondary immune response. For the secondary immune response, because they store information about the antigen fought or killed by the immune system in the past. Then the duration of this primary immune response, it is a slow one because it enters for the first time, so they must recognize it at first and then make antibodies and kill it, infect it and attack it and fight and then kill it. So this is a slow response. The first takes place from 5 to 10 days. That is because these cells need to multiply and to make a defense for it. while in the secondary immune system, immune response, a very fast response to the pathogen, which will be destroyed before the appearance of the symptoms. Because this one in secondary, everything is ready, so it attack it immediately and there is no symptoms appear. While in the primary, some symptoms or the symptoms may appear, or this person may be ill for a while. During this time of the incubation period, or the 5 to 10 days, during this time the infection could be widespread and the symptoms of the disease appear because the infection would be widespread. But here in the secondary one is not accompanied by the disease symptoms because it is destroyed very fast. The primary immune response, we found that the memory cells are formed during this one and stay in a dormant state or incubate state, while the memory cells become active in the secondary immune response and response to the pathogen once it enters the body again or another time. The memory cells. Then what is a memory cell? Is a type of cells which store information about antigen fought by the immune system in the past, or fought or killed by the immune system in the past. The memory cell has two types, the memory B cells and the memory T cells, and they are characterized by produced during the primary immune response. Memory cells can live for tens of years and may survive till death, but the B and T cells can survive only for few days. The B cells and T cells, not memory B cells, memory T cells. Memory B cells, memory T cells, they can live or stay for lifetime. But these B and T cells only, they can be can survive only for few days. During the second infection with the same pathogen, the memory cells respond to the pathogen once it enters the body, where they stay to divide quickly to produce large amount of antibodies and activate T cells within short time. So this is the characters of the memory cells. First question. Write the scientific term for each of the following statements. A type of cells which store information about antigen fought or killed by the immune system in the past. Yes, it is the memory cells. Another scientific term, the response of immune system to the infection of the same disease again. Secondary immune response. Very good. The resistance of the body to pathogens that infected the body before. Acquired immunity. The immune response done by T lymphocyte, which is done by the receptor found on its membrane that give it a specific response to antigen. Cellular or cell-mediated immunity. A series of specific defense mechanism that the lymphocyte perform to resist the pathogen. It is the immune response. Then what about the comparisons? Look for these two comparisons. Compare between acquired immunity and natural immunity, primary and second immunity response. Let's begin by the natural immunity and acquired immunity. The natural immunity, it is a group of defense mechanisms that protect the body, while the acquired immunity, the resistance of the body to pathogen that infected the body before. In the natural immunity, non-specific or innate immunity, while the acquired, they are specific or adaptive. Natural immunity do not make memory cells. The acquired immunity form memory cells. The first line of defense includes the skin, mucus, tears, sweat, and hydrochloric acid of the stomach, secreted by the stomach. The second line of defense, inflammatory response, represent the third line of defense, and it is subdivided to two mechanisms: humoral or antibody mediate immunity, cellular or cell immediate immunity. The second comparison, primary and secondary immune response. For the primary immune response, the response of the immune system to a new pathogen. In the secondary, it is the response of the immune system to the infection of the same disease again. In the primary, the B and T cells are responsible for the immune response to the pathogen and attack it until it is destroyed, while the memory cells are responsible for a secondary immune response because they store information about the antigen killed by the immune system in the past. Primary immune response, slow response. The first takes between 5 to 10 days. That is because these cells need to multiply, while the very fast response, the pathogen will be destroyed before the appearance of the symptoms. During the primary immune response, during this time, the infection could be widespread and the symptoms of the disease appear because the infection would be widespread. While in the secondary one is not accompanied by the disease symptoms because it is destroyed very fast. In the primary immune response, memory cells are formed and stay in the dormant state or incubated state, while the secondary immune response, the memory cells become active and respond to the pathogen once it enters the body again. And now, my dear students, we finished the chapter 4 of immunity, and next time we are going to review all of this chapter. See you next time.